Imagine a warrior who, on realizing that his continued existence would dishonor his clan, chooses to end his life with dignity, not out of weakness, but as an act of honor and protection. In Japanese tradition, this is known as harakiri, a ritual suicide performed with precision, purpose, and a deep sense of duty.
Now shift your gaze from the battlefield to the microscopic world inside your body, where cells are engaged in a similar practice. When a cell detects that it's damaged, infected, or simply no longer needed, it doesn't wait for destruction from outside forces. It initiates its own self-destruction, a highly organized process known as apoptosis. Extrinsic signaling happens whenever an external threat is detected through special receptors called death receptors. These are part of the TNF receptor superfamily and can include FAS, TNFR1, trail R1, R2, also called death receptor 4, 5 respectively. Through a particular death domain, they help in interaction with adapter molecules who are in turn attached to procaspase 8. This whole complex is what is called DISC and will ultimately lead to activation of the executioner caspase. But more on that later. Internal signals can include damage to the DNA, oxidative stress, microtubular alterations, or even accumulation of misfolded proteins. Signals in the mitochondria are perhaps one of the most important and is eventually due to changes in the membrane permeability. Another important signal is DNA damage. Base pair mismatch is picked up by P53, which will then activate pro-apoptotic signals while downplaying the anti-apoptotic ones. Ultimately, it results in changes in mitochondrial permeability. The role of the transporter MPTP is also mentioned here. Contents from mitochondria like cytochrome C will then be released, which will interact with APAF1 and ATP to form apoptosome. This will then interact with procaspase 9 to get activated. The end result, executioner caspases are activated. Now you would have noticed how in both extrinsic and intrinsic pathway, executioner caspases are ultimately activated. They are a group of protease and on cleaving gel solule will disrupt the cytoskeletal structures. This will manifest as cell shrinkage and there will be blebbing of the membrane. The protease will also cleave cytoplasmic proteins and the cell will start turning deeply eosinophilic. The protease also cleaves the nuclear proteins, resulting in the eventual loss of the lamina. This will start the process of chromatin condensation. This step is called pyknosis. The executioner caspase will also enable the action of caspase activator DNase by cleaving its inhibitor. Once the dissolution of the nuclear membrane happens, the endonuclease breaks up the DNA into short fragments. This is what we call karyorrhexis. The cell then fragments into apoptotic bodies during the budding phase. They contain cytoplasm with tightly packed organelles with or without nuclear fragments. They are then phagocytosed by the surrounding cells like macrophages, parenchymal cell, or neoplastic cell. The macrophage that engulfs the apoptotic body are called tingible body macrophages, and the word tingible refers to nuclear debris from the apoptotic cell. Here are some live videos that you can take a look at in order to revise the steps. Let me know in the comments if you can identify the stages as they happen. And these are some of the differences between apoptosis and necrosis. Don't forget to like and share with your friends, and do subscribe if you're interested in more medical content.